Dear learners, welcome back to NPTEL online certification course on novel drug delivery system. I am Dr. Satish Davanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our novel drug delivery systems, we are discussing the topic, that is scalability and regulatory aspects of novel drug delivery systems. So far we discussed on the various topics which are related to the important significance of novel drug delivery systems, and then further we discussed on the challenges, issues of the nanoformulations. Further we discussed on the scale up issues, and then we discussed on the regulatory issues. In last class we discussed on the what are the regulatory bodies, and then we discussed the role of regulatory body in the drug discovery development, and in that we discussed different stages of drug discovery development. At each stage we have regulations need to get the approval from the regulatory body, and we learned about INDA, investigational new drug application, which is required for a human clinical trials of any new drug. NDA is for the new drug application for marketing and authorization of new drugs, that is given by the FDA after reviewing of all clinical data. And yes, we learned about BLA, biological licensing approval or application. So again, that is for the biological molecule which are different from small molecules which are drugs. Now then we have seen about ANDA, that is abbreviated new drug application, which is for the those drugs patent is expired, such drugs can be again translated or converted into the generic molecule. Today we are going to discuss on some of the challenges associated with the nanomedicine regulations. So here you can see on the screen the summary of the major challenges associated with the nanomedicine regulations. The first one is lack of unfiled. The first point is lack of unified definition and the classification of the nanomedicines and nanomaterials. That is one of the problem. So we use multifunctional carriers and there is no proper definition so far, because theoretically we say that 1 to 1000 nanometer, right. And now you can see that, so when you are taking orally and when you are going for IV, so the definition is not going to work, right. So we prefer the 10 to 100 nanometer or 200 nanometer for the intravenous or some for parenteral applications, whereas oral we can use 500, but there is no defined definitions as well as classifications of nanomedicine and nanomaterials so far, but still we discussed in our second week definitions of nanomedicines and nanomaterials. So that is one of the major problem which is we are facing in the challenges in nanomedicine regulations. Lack of agreed regulations, nothing but, so when you compare with the conventional formulations like tablets, even with the oral formulation tablets, capsules and syrups and other types of formulations, even the topical, we have some of the correlation, some of the agreement with with other countries. But whereas for the nanomedicines, we do not have proper agreement in one within the country as well as within some region and within the world. So that is one of the major issue we are facing here. You can consider like a dissolution apparatus, like we have some standards even in for the processing, even the manufacturing and GMP guidelines we follow, and even the test results also we need to perform it particular as per the some pharmacopoeial guidelines. But here we do not have some agreed regulations, like similarly we use like ICH guidelines for pharmaceutical products. So, this is a harmonization of the Japan, US as well as Europe. So, from that we need to give some technical documents for the registration of any pharmaceutical products. So, this agreement, this is a harmonization between the multiple regulatory bodies similarly, which is not there. Now, the third one is analytical methods differ for each nanomaterial. Again, so this is a lot of issues, because we have different nanomaterials, that is polymer is there, and again we have a lipid and we have a inorganic materials, right. And again in a inorganic we have so many materials. We do not have the standardized methods for each nanomaterial. So, that variation is again one of the major problem in the nanomedicine. Now, pharmacokinetic profile diverge from standardized constituent material. Now, we are taking in a simple one material, that kinetic profile is completely different from when you are coming as in with an entity or with the formulation. So, that profile is completely different, and again that is very challenging for understanding the role of material in the pharmacokinetic profile. So, when you are modifying your formulation, your kinetic profile will change. And again the fifth point is stability issues after scale up for manufacturing. Now, yes, we have the luckily we have ICH guidelines that will help us to see the stability problems by putting your formulation into the controlled environment as well as in the temperature as well as in the relative humidity, we will understand. But again after doing of the scale of formulation, we will face lot of stability issues, that variation comes during the storage, during the process, even the instability issues of your formulations. So, that again one of the biggest issue. So, the guidelines are need to be revised and need to be developed for the stability standards. And again current in vitro preclinical toxicological studies fail to mimic in vivo complexity. So, this in vitro in vivo correlation, right. So, that is also one of the major problem. So, the toxicological studies will not if your molecule is getting safety in the in vitro studies or preclinical studies, and then further if you want to perform in the human beings, and you will see lot of toxicity. So, that complexity need to be avoided, need to get some right models or prediction tools or some of the softwares which may help us to understand the complexity of the molecules in the animals as well as in the human beings. So, that is also one of the major problem. And systematic biodistribution and fate, and yes, when you are taking this formulation, like their physicochemical properties may vary when you are going from in vitro to in vivo studies. Like we use simple example like SMEDs, small micro emulsifying drug delivery systems. When you want to we make this formulation by using of oil, surfactants and co-surfactant, we do not add water in this formulation. What we do, we take this formulation, we put it in a 0.1 normal HCl or other condition with the like simulated gastric fluid, and at some RPM we will run and we will do the dissolution study, and we believe that it forms the micelles. So, with the size range of around 10 to 100 200 nanometer, we will get this. And yes, sometimes these may not happen in the in vivo conditions, because of in vivo conditions different, there you have lot of physiological alterations may happen, even food also may interfere, gastric motility condition also may interfere, even some enzymatic also may leads to alter your biodistribution and fate of your molecule. Now, the chances of the aggregation, precipitation of the drug may be high in terms of in vivo applications. So, that type of studies need to be performed. And possible environment as already we discussed, harsh acidic conditions or some the enzymes which may degrade your molecule. So, that is also one of the problem, because in nanomedicine we are taking nano, our size and shape and charge are very important. If in an environment it undergo aggregations or some precipitation happens, then it will not fulfill its criteria. So, that is why we need to understand all these issues by using of the right techniques and with the understanding. So, now here is one more consideration. If you want to make any compliance within the regulatory body, so the quality of pharmaceutical products should be designed and built during the manufacturing process. Now, the technique is QBD, quality by design. We have the conventional process, that is QBT, quality by test. By doing the test of the raw material, by doing the test of your finished product, we will judge the quality. That is actually problematic, and it will waste lot of time and energy and manpower, right, even the cost also. So, quality by test is will prove after doing the test, but the new regulation suggests that you have to practice this quality component during the design and the build during the manufacturing process. With that, we can achieve QBD, that is a systemic approach to development that begins with the predefined objectives we target initially based on the designing approach and emphasizes product, process understanding and the process control based on sound science and quality risk management. We try to identify what is the risk and how to overcome and how to replace, how to overcome completely from the process. So, these are called as quality by design approaches. Now, QBD is one of the most important initiative by the US FDA, that pharmaceutical quality for the 21st century. A risk based approach in 2002 by FDA was the first step towards the goal of QBD compliance. Now, most of the ICH guidance also will recommend to implement the QBD, because that is the goal of the ICH is to maintain the quality, safety and efficacy. So, in all these cases we need to maintain the quality by using of the QBD approach, which is the driving force for the company. If you implement, the chances of getting approval and compliance is very high for your product and for the process. Now, these are the some elements we need to know, right. So, total 4 elements in the QBD, that is QTPP, nothing but quality, target, product, profile. Second is CQA, critical quality attributes, CMA, critical material attributes, CPP, critical process parameters. So, 4 elements are very important. All these elements are collaborating in step by step approach to draw framework of the QBD, nothing but when you are implementing you will get the quality. The recruitment of these key elements in the QBD method needs well defined experimental design combined to proper statistical analysis. From that you will get in a design space or design experimental design. With that you can develop QBD profile. Now, you can consider here the QTPP, quality target profile, nothing but you can read here with me, including dosage form, delivery system, dosage strength. It is a prospective summary of quality characteristics of the drug product to be achieved at taking into dosage strength and container closure system of the drug together the attributes affecting pharmacokinetic characteristics. So, it speaks about the quality of your final product. It is speaks about the what type of dosage form, what type of delivery system, what dose strength you need, and the routes of administration, and also speaks about the product quality, that is sterility, purity, and appropriate intended to use it for the market. So, that is quality targeted product profile. It is the broad term of your dosage form and delivery system and dose strength. Now, CQA is critical quality attributes, nothing but it speaks about the quality of your output, that is nothing but finished product, about the physical, chemical, biological properties of your material, right. So, there you need to identify what are the critical quality attributes for your product, what are the range that need to be identified. Now, CMA speaks about the material is nothing but input material. When you are giving input and process, you will get an output, that is CQA. Now, we will see that also, right. CMA is nothing but again physical, chemical, biological, micro properties of your input material, and you have to again identify what is the range and distribution and limit of that material. It may be drug substance, excipient, and in process material. Now, CPP is nothing but parameters monitored before or in process that influence appearance, impurity. That means process parameters we need to identify. Not all process parameters are important for your method or your product. We need to identify what are the critical process parameters, how much and at what range, that is important. And now you can see ICH also suggests Q8 guidelines, that is pharmaceutical development, Q9 risk management, Q10 pharmaceutical quality system. This all will help to design the QBD, industry requirement, and from patient and also from the regulatory body, that is a product quality life science. So, here you can see QTTP, QCA, critical quality attributes, that is to determine the potential critical quality attributes, and again risk assessment also very important, that will give the relation between your input, process and the output. And the design of experiment will help to design the understand the design space, at what ratios we can work out. So, that will give the various statistical approaches and the design space. From that you will get the design space, from there we can generate molecules or generate products and we can test it. So, control strategies, continuous improvement and the drug life cycles, for that also you have to understand. Now, you can see here step by step, once it is defined, you can also define what are the critical quality attributes, and based on that risk assessment experimental design, you can identify again input materials, what are the range, and then process production, critical process parameter, then you will get the output. So, these are the settings are important, critical quality control testing also very important to see the quality of your product. Now, you can see some example here. So, try to understand this is the input material, you are processing and you will get the output, okay, pharmaceutical unit. Now, you can see here, if I want to go for the immediate release, okay, here is my material attributes, here my process parameters. Now, you can see there are the so many grades and so many variations are there, but all are not important. From that you need to take critical material attributes. For this drug, which is a critical? PSD, particle size distribution, polymeric form, that is enough, right. So, need to identify your critical material attributes and also critical process parameter, not all are not important, you need to identify. Then process with the DOE and scale up equations in both case, and just do the processing and identify what are your the critical quality attributes, that your outcome tablet, how much assay you want, contain uniformity, dissolution, stability, that you need to identify, that is called as CQA. Now, you can see here again with this diagram, you can see on the top you will see critical material attributes, and in the below you will see critical process parameters and the critical quality attributes. Now, in the materials you can see the conditions are important, and polymer concentration, type, excipient, that materials also important, and their properties also important. Now coming to the process, what are the condition, which method and how it works, type of construction and other arrangement accesses are important. From that you will get the attributes, size, PDI, morphology and other formulation. These also important need to be. If you want to know, you can read here. And again this is help for the optimization of the nano formulation. Just within one example here I have mentioned, you can read here. So critical quality attributes to be considered to support the drug's target product profile. So selection of CQA will help for the achieving the QTPP, right. So dosage form important, that you need to identify, dose strength, stability, kinetics. From that you will design particle size and distribution, zeta position, drug content. And risk assessment is important, and pre-formulation study and taking of updated risk assessment, yes, by implementing of the design of experiment, box Benken design, establish product design space, and again from that you will get a full factorial design, and then you can optimize your formulation. With this you can achieve the quality of the product by designing. Now again you can see here the drug attributes, nano system attributes, dosage form attributes, desired formulation attributes. Now we can see here, so these are the solubility, crystallinity, polymorph, hygroscopic, size, surface chemistry and excipient properties, and again you can see the various desired properties. With that you will achieve the quality, safety and the efficacy of the formulations. Now this is one important considerations when you are you can go through into the, you can type the guidelines for evaluation of the nanopharmaceuticals which are provided by the Indian government with the help of various societies. And when you go into this, and you will see lot of definitions which are already discussed in our first slides. So lack of definitions and the classification, that is one of the major challenge in the regulatory. So now Indian government took stand and they prepared and classified all this information. Now you can see, if you open this document, you will see all this information, abbreviations and the introduction, scope of guideline, general consideration. Now we can see here the nano pharmaceutical definition, characterization, scientific rationality, and again you will see what type of test need to be performed, and then how the data need to be informed. Just I will show the summary. So this is the guidelines you can see here. So these are the, you can see various introduction and scope of guidelines, and you will see lot of definitions also here. So that will help for you understanding how this will difference from other regulatory bodies. Now you can see the size. Now you can see 100, 1 to 100 nanometer, and these are the about the particular distribution. See, now you can see categorization here, right, and also you can see the definitions according to the degradability of nanomaterials, and you can see the inorganic and organic materials. So you can go through these, I will upload in the final materials, and that will help for your further studies. You can see lot of stability and animal pharmacological data also is provided, and what type of information need to be provided for the, if you are going for any, yes. So this information need to be provided for the any type of novel drug delivery or nanomedicine systems, and all data need to be characterized by using of these instruments. So you can see some stability issues, stability related document also, pharmacokinetic and pharmacodynamic. So Indian government also is helping to overcome all such issues in the regulatory body. So in our future direction we can, yes, clinical translation is much important. That means moving from lab scale to large scale is very, very important. Again moving to the clinical site also is very, very important. So that need to be mitigated by defining stringent criteria, that means proper testing as well as control checks during the nanomedicine and development. And yes, biocompatibility immunotoxicity studies need to be evaluated through proper characterization and by using of pharmacotoxicological studies. And yes, for the defining of therapeutic index, dosage regimen, maximum tolerated dose and the route and target of administration is very, very important. And yes, drug hemolysis, complement activation, cytokine release, opsonization, phagocytosis and PK study, pharmacokinetic studies that should represent just part of in vitro and in vivo data correlation required for the clinical translation of the nanomedicine. And there are few nowadays technologies which is also help for the overcome the all type of scalable and regulatory approaches. That future is now nothing but already we have discussed today in this week about the continuous manufacturing techniques like microfluidics, as well as we learned on the HME and then high pressure homogenizers. Such continuous manufacturing can be helpful, and not only that, artificial intelligence and machine learning also will help to overcome all the process controls and the problems which are going to happen with the in the complexity in the manufacturing, as well as complexity in the body and the complexity within the nanomedicines and the novel drug delivery system. Regulatory harmonization is very, very important, like we have ICH guidelines, that is International Council of Harmonization for technical requirements of human products for the registration. So, such ICH guidelines are very, very important. So, it is a harmonization which brings industry people as well as the regulatory people. So, they will come and discuss and make the guidelines accordingly. So, AI and ML formulation process control, which from that we can predict the modeling and optimize the formulation variables, and we can also get automated data analysis of quality control. Real time release and real time quality assurance need to be maintained. A future where products are released based on continuous real time data, not on end of the process of batch testing. So, by using of such techniques or some digital process controls, with that we can achieve or we can overcome all type of scalability and the regulatory issues in the novel drug delivery systems. So, with this we are going to stop, we are end up with the coursework. So, so far we learned various aspects of the novel drug delivery systems, and you can refer these notes for your further reading. If you have any doubts, you can interact with the discussion as well as live session meetings also. We are happy to help you, and once again I wish good luck to everyone, and I also convey my regards to everyone for attending and also joining this course. Good luck to you all. Thank you so much.